Good morning, and thank you for the opportunity to speak today at BioKorea 2020 on global cooperation in the COVID-19 vaccine development process. Who would have thought, looking back to Christmas of 2019 or to the New Year of 2020, that we would be in the middle of one of the greatest pandemics that this century has ever seen? Worldwide, by the 7th of May 2020, 3.8 million people had been infected by COVID-19, and there were 265,084 deaths in 187 countries around the world. But COVID-19 is just the latest pandemic. It is not the first or the last. This is a cartoon reminding us of the ability of the pandemic that we call AIDS to strike anywhere across the world with a network, and its only purpose is to infect and kill. But this dates to 1983. The world has seen the Black Death, or the bubonic plague in 1562. Smallpox has been around since 3,000 years before the Common Era, until it was eliminated in 1979 by vaccination. Cholera, a disease that IVI has worked on since its founding, has been involved in 7 global pandemics. We are in the middle of the 7th pandemic today. The Spanish flu was the last great pandemic, going around the world in 1918 to 1919, killing millions of people. And of course, we have COVID-19. Not to forget that although Korea appears to have won the first battle in the war against COVID-19, there will be other times when Korea will have to face outbreaks of COVID-19, as it has just seen in the district of Itaewon in Seoul last week. Coronaviruses are RNA viruses that infect mammals. And coronaviruses are what we call promiscuous. That is, they can use more than one mammal as their host. So you can see in the bottom right-hand corner the fact that a coronavirus involved in SARS initially infected bats, and then palm civets, a type of cat-like creature, and then from palm civets to humans. The coronavirus known as MERS, which was here in 2015 in Korea, started in bats, infected camels, and from camels infected humans. What we call SARS-CoV-2, or COVID-19, or corona, as it's called in Korea, started in bats and probably involved an intermediary host called the spiny anteater, or pangolins, before infecting humans and causing the pandemic going around the world today. As we develop a vaccine, there are some important questions that we would ideally like to have the answer to as we proceed with vaccine development. The first and possibly the most important of these is, does infection provide immunity from reinfection? Now, why is this important? Well, in a classic vaccine model, say hepatitis A or polio, almost all people who are infected run the course of the disease, whether that's the development of hepatitis or not the development of hepatitis. But in the end, that person is protected against further reinfection with hepatitis A, or if they had polio, from reinfection with polio. This means that the body's immune responses, the development of antibodies, cytotoxic T cells or helper T cells, was sufficient to control and eventually eliminate the viral infection. This means that typically then, if we could develop a vaccine that could mimic those responses, then the vaccine would likely, probably, be able to prevent infection. And that's what we eventually test in phase 1, phase 2, and phase 3 trials in humans. The second important question is, which of those immune responses is the most relevant? Is it the antibody production? Is it the helper T cells or the cytotoxic T cells? And again, why is this important? Because the design of the vaccine really dramatically affects the kinds of immune responses that will predominate. So for instance, DNA vaccines may make pretty strong cellular immune responses, but antibody responses may be lower than is seen typically with a subunit vaccine. Changing the adjuvant from alum to AS01 or AS01B, which is a GSK adjuvant, may improve the cellular immune responses against an antigen present in the vaccine. So again, what immune responses do we want to target, and how are we going to get those correct immune responses in the ultimate vaccine? The next question, and this is one that we ask all the time and are asking today about COVID-19, is what is the best animal model? Of course, animal testing often begins in mice because they're small, they're cheap, they're convenient. But in the end, are mice the appropriate animal model to take a vaccine forward into human clinical trials, where the cost and complexity are a big issue? So as you see in the picture, there's a monkey holding a rat. And what animal model do we actually use? Do we use mice, rats, ferrets, or monkeys, or a combination of all of them? And as is commonly said in vaccinology, mice lie, monkeys exaggerate, only humans tell the truth. So again, we'll know the answer to this in human clinical trials, but hopefully the animal models will help us to refine the vaccine in order to bring forward the best vaccine for human clinical testing. The final question, and one that is really one of the most important that we will deal with, is the vaccine safe? And what's shown in the picture is actually a reaction among people in the 18th century to the process known as vaccination, the initial use of cowpox vaccine to prevent the disease known as smallpox. And in those days, people who were being injected with the cowpox vaccine for smallpox were afraid that they would develop little cows springing out of their arms, and that's what the cartoon shows. However, we know that this cowpox vaccine was instrumental in eliminating smallpox from planet Earth. So in 1979, the use of that particular vaccine was associated with WHO's declaration that smallpox had been eliminated from Earth. And it was very important. So although safety is an issue, efficacy, the ability of vaccine to prevent infection and disease, is really another part of the equation, and critical part of the equation. We're often asked how long it's going to take us to develop a vaccine against COVID-19. And we always say, well, under normal circumstances, it will take 5 to 10 years. Under extraordinary circumstances, and I think the COVID-19 pandemic qualifies as an extraordinary circumstance, we say 6 to 18 months. Now, how will this happen? Under normal circumstances, again, with the 5 to 10 year timeline, it costs between $500 million and $1.5 billion to develop a vaccine. And the failure rate of vaccines going from the laboratory into humans and finally into final phase 3 testing and approval by the Food and Drug Administration or the Ministry of Food and Drug Safety has a failure rate of 93%. That means that the complexity and cost of vaccine development at $500 million to $1.5 billion, coupled with a failure rate of 93%, makes the process of developing vaccine a risky one for companies. And again, this informs a lot of the structure of the clinical trial design and the delays that are put into place in order to really know whether a vaccine that is moving forward is the correct one, is cost effective, can be manufactured. All these things are things the company is thinking about when it has 5 to 10 years to develop a vaccine. But given the extraordinary pressure and the urgency of the COVID-19 pandemic, we've said that it will probably take 6 to 18 months. And how is this going to be accomplished? So again, we will go through the same stages of clinical testing. We do phase 1 trials typically in about 50 people, looking at safety. We'll do a phase 2 trial, several hundred people, looking at what we call the target population, the group of people in whom we will ultimately use the vaccine. And we look for immunogenicity. Is the vaccine making the correct immune responses? Finally, in phase 3, we'll look at efficacy and safety. Again, the combination of the two being the criteria for moving a vaccine forward for licensure. Instead of having, though, 5 to 10 years, we're going to have to telescope phase 1, phase 2, and phase 3. So we're starting to see, and you'll see that on a subsequent slide, phase 1/2 clinical trials, where phase 1 and phase 2 are merged together, and we're overlapping phases. So that in the end, we'll see the same efficacy signal, but the safety signal may be a bit smaller than we're used to seeing. And so we will have to follow up people for longer periods of time to ensure that vaccinated individuals, in addition to being protected, don't suffer some unknown effect later on at 3 or 4 years after they're vaccinated. But again, the goal here is to try to accomplish something that normally takes 5 to 10 years in 6 to 18 months. If you look at the slide, on the far left, you see a schematic, a cartoon of of the coronavirus, of SARS-CoV-2. And the blue protrusions, the club-like things sticking out of the virus, are what we call the spike protein. The spike protein is very important because we know from other coronaviruses that antibodies against spike protein appear to neutralize the virus, bind to the virus, and keep it from infecting human cells. That's really important because that's one of the things we'd like the vaccine to do. So if you look at that subunit, and in the next figure over, you see a trimeric. It's a homotrimer. It's three spike proteins put together, actually form the spike on the viral surface. And we have to be able to duplicate that because that's what we want the antibody and cellular responses to recognize. So how are we going to do that? You could make an RNA or DNA vaccine, so nucleic acid vaccines, that are quick to make, but currently we don't have any licensed RNA or DNA vaccines. We could make a spike protein itself using cell culture, and that would be similar to other commonly available vaccines. We could just take the vaccine, the spike protein, the genetic information, and insert it into a viral vector. We could put it into MVA, modified vaccinia ankara, or into adenovirus 26, or into the Oxford vectors, which are simian adenoviruses. Or, as an alternative approach, we could just grow up tons and tons of virus, millions and billions of viral particles in cell culture, and kill the virus using heat or formaldehyde or some other commonly used approach. And this is, probably one of the simplest forms of vaccine production. It's relatively inexpensive, but it has some complexities because SARS-CoV-2 is something that has to be handled under very strict biosafety and biocontainment procedures. So in addition to making several 100 million doses a year of this vaccine, you'll have to put into place appropriate levels of biosafety containment. The final kind of vaccine is one that would be great if we had it because live attenuated vaccines, vaccines that are still alive and can still replicate but do not cause disease, are probably the vaccines that give you the most durable immune responses and the most substantial protective immune responses. But because of safety concerns, they do take longer to develop. So ideally, it would be great to have one, but it may take a bit longer than it will for the other vaccine approaches. And you can see the different kinds of vaccine approaches here. With a number of stated programs available for each of the vaccines for SARS-CoV-2. And as of the 30th of April 2020, there were over 100 programs in COVID-19 vaccine development. So imagine starting in January, not knowing anything about this. By the end of April, 4 months later, there were over 100 different vaccines in the clinical development pipeline, and 8 of those vaccines had already entered human clinical trials. This really is unprecedented in the history of vaccine development for us to move this many vaccines for a brand new, for what we call a novel pathogen, forward so quickly. Going now into how we're going to do this. Epidemics are very expensive, and the World Bank estimated that the cost of outbreaks in the 21st century would be $1.6 trillion. MERS cost the Republic of Korea $10 billion. SARS cost East Asia $40 billion. Ebola cost West African countries about $6 billion. The SARS-CoV-2 pandemic is already into the trillions of dollars, so the original World Bank estimate is low. And we don't know the total cost in dollars, and we certainly don't know the total human cost and societal cost of the outbreak. But one of the things that became clear during the Ebola outbreak is that we can never develop a vaccine solely during an outbreak. Because what happens is, during the outbreak, people get very nervous and excited. Governments put money into vaccine development. Governments like President Obama called the heads of major companies and asked them to drop everything and make a vaccine against Ebola. But within 9 months, the epidemic was waning, and interest in developing an Ebola vaccine dropped. Luckily, one vaccine was able to be tested and was shown to be effective against Ebola. But the other vaccines didn't make it into clinical testing, and as a result, companies lost hundreds of millions of dollars in productive time spent working on other things. Recognizing that this was a problem, a group of individuals proposed something called the Coalition for Epidemic Preparedness Innovations, or CEPI. And in its brief lifetime, Since being announced in January 2017, CEPI has amassed over $1 billion in funding from countries all over the world, including the Republic of Korea, and from foundations such as the Bill and Melinda Gates Foundation and the Wellcome Trust. And it's put this money to use developing vaccines for outbreaks, knowing that you had to have an organization that was capable of funding the development of a vaccine through what we would call a stockpile, so that we had tested it in humans in phase 1 for safety and phase 2 for immunogenicity. And if the outbreak isn't present, then we would stockpile a certain number of doses of that vaccine just in case. And if an outbreak occurred, we could very rapidly test that vaccine to see if it was safe and efficacious. CEPI also prepared for something called Disease X. And that would be an unknown outbreak, an outbreak of an unknown disease. What would we do? Would we have funding available? And in fact, COVID-19 fit the pattern for Disease X. So shortly after the sequence for COVID-19 was published by the Chinese in the middle of January 2020, CEPI put out a call for proposals. Who can get a vaccine into humans in 4 months? And 4 groups received initial CEPI funding. Moderna, which is an American company that makes RNA vaccines, Inovio, an American company that makes DNA vaccines, CureVac, a European company that makes RNA vaccines, and the University of Queensland, which had a very unique protein vaccine approach. Of those 4 initially funded programs, 2 of them are entering phase 2 of clinical testing. So in 4 months, they've gone from nothing to actually being in the phase of vaccine testing where we're looking to see in the target population whether the vaccine is developing appropriate immune responses, protective responses against COVID-19. Again, an amazing tribute to an organization that really didn't exist until 2017, but has stepped forward as a global solution to a problem that threatens us all. There has been unprecedented collaboration in the COVID-19 pandemic. The World Health Organization, whose role is to bring people together and to come to consensus on diseases that threaten all of us, initiated the Solidarity Drug Trial in over 100 countries with 4 drugs that might treat COVID-19, including remdesivir, hydroxychloroquine, lopinavir, ritonavir, and beta-interferon. Solidarity also exists as a vaccine trial using something called an adaptive design, which allows multiple different vaccines to be compared. So far, none of the vaccines have entered the Solidarity vaccine trial, which is really designed to handle what we call phase 3, the efficacy trial, the trial to see whether the vaccine protects against infection and is safe. In addition, the regulatory agencies around the world, the organizations that are responsible for ensuring that vaccines are both safe and effective before they're licensed for sale in countries, so the US FDA, the EMA, the Ministry of Food and Drug Safety, and others, met in March of 2020 under the International Coalition of Medicine Regulatory Authorities, ICMRA, to come up with a series of guidelines to companies to really help the companies to understand what the regulatory authorities would require in order to accelerate vaccines into human clinical testing. So what things could companies do in order to facilitate human clinical testing? And some of those things were, it would be helpful if there were preclinical data or a lot of previous human data using a similar platform. So for instance, the Inovio vaccine has been used for multiple studies in animals and also has been in clinical testing for different diseases, not necessarily for COVID-19 at that time, but it had been used as a MERS vaccine. It had been used as a Zika vaccine. And so safety data indicated that the Inovio vaccine, the platform, the DNA vaccine platform, was safe. So this allowed the US Food and Drug Administration to allow them to skip one of the steps, which was an animal toxicity study. Now, it turns out that the FDA allows DNA vaccines to move forward that way anyway, but this saves months in terms of the time it takes to go from the initial design of a vaccine in the laboratory through the initial stages of animal testing and then into humans. So using these mechanisms, the different regulatory authorities set forth a series of guidelines that would help companies to understand what needed to be done in order to advance vaccines very quickly into human clinical trials. CEPI, again, the Coalition for Epidemic Preparedness Innovations, and the Brighton collaboration convened a workshop to look at a very specific safety issue that may have been present for SARS-CoV-2 vaccines, and came up with a series of recommendations on how to move forward in order to evaluate and ensure that vaccines that are developed for COVID-19 do not have any future safety problems. Finally, the Bill and Melinda Gates Foundation is exploring ways that it can facilitate and boost manufacturing capabilities so that, if you imagine how many people will need a COVID-19 vaccine, when we have a safe and effective vaccine, it can be manufactured in the quantities and quality sufficient to vaccinate all the people in the world who need the vaccine as quickly as possible. Another thing, and a similar organization exists in Japan. The Korean government created the Research and Investment in Global Health Technologies Fund, which is a collaboration between the Ministry of Health and Welfare, the Bill and Melinda Gates Foundation, and pharmaceutical companies and vaccine companies in Korea. And they put together funding in order to work together on vaccines for global health. And we're hoping that GHIT, so RIGHT and GHIT, the Japanese version of RIGHT, will fund vaccine development for COVID-19 as well. Again, illustrating the nature of the global efforts that are going into solving a vaccine, finding the vaccine solution for the SARS-CoV-2 or COVID-19 pandemic. This is a list of the vaccines that are currently in phase 1 and phase 2 testing in humans. And you can see there are 8 different vaccines, and I'm not going to go through all of them. Some of them are adenovirus vaccines. A Chinese company, CanSino, has an adenovirus type 5 vaccine. The Oxford Group has a vaccine based on a simian adenovirus. 2 Chinese companies have heat-inactivated whole virus vaccines. Some of those vaccines have been shown in monkeys to be protective. Again, that's great news. The Inovio vaccine is in phase 1/2 testing in the United States, and hopefully phase 1/2 testing will start in Korea by June. And the Moderna vaccine, which is an RNA vaccine, a collaboration between Moderna and the U.S. Vaccine Research Center, is ongoing as we speak. This is a quick view of the draft phase 1/2 clinical trial that we hope to start in Korea. It's a collaboration, again, an international collaboration with CEPI, Inovio, the Korean NIH, and IVI. We're often asked the question, now remember, we're trying to do something in 6 to 12 months that would normally take 5 to 10 years. Are you going to cut any corners? And I think that the bottom line is this: vaccines should be safe and effective. There will be an independent data and safety monitoring board. These are routine in vaccine trials that are done under the old timeline, 5 to 10 years, but they will be mandatory in vaccine trials that are going to be conducted under the accelerated schedule also. Data and safety monitoring boards are able to look at safety information and efficacy information in real time and can advise the sponsor of the trial whether the trial can be safely continued or needs to be stopped. So looking at the different components, in the 5 to 10 year system, or in the 6 to 18 month system, we will have an efficacy signal, does the vaccine work or not, by 12 months. Whether the vaccine continues to work at beyond 12 months, we won't have under the 6 to 18 month system, but we usually do under 5 to 10 years. But the people who design the efficacy trials under 6 to 18 month accelerated schedules can incorporate additional time points, even though they won't be a part of the initial approval of the vaccine. They can be a part of the longer term follow-up designed into the protocol by the company or organizations designing the trial. Will there be a data and safety monitoring board? Yes, under both. And those data and safety monitoring boards will review the trial for safety, for something called futility. Is the vaccine trial able to accomplish what it set out to do? Or sometimes they do interim analyses. So if the vaccine is working so well that the placebo group is at some perceived risk, then a trial can be stopped early for efficacy. The final question has to do with safety. And so because these trials are going to be conducted within a 6 to 12 month time frame, we will only have safety data at the 12-month data collection point. And likely if there is a vaccine that works, it will be licensed with only 12 months worth of safety data. That does not relieve a company or the sponsor from continuing to follow people for safety. And it is a best practice, and it's done routinely in other vaccine trials that are 5 to 10 years, to watch people for longer periods of time, again, to ensure that people who participated in vaccine trials remain safe and do not undergo any additional risk as a result of their participation in the trial. And this is, again, a routine procedure that is followed by vaccine companies around the world as a best practice. Now, being at the International Vaccine Institute, we always think with the end in mind. And the end for us, for any vaccine, is impact. How many lives are saved? Does the vaccine decrease disability-associated life years? But more important than that, which is a direct effect of vaccination, are the indirect effects of vaccination also accrued? Will we have healthier families? Will this decrease poverty? Does this improve cognitive and physical development? Does this improve education? Does it improve economic growth? And for for a COVID vaccine, the consequences and impact will be even greater because of the threat that COVID produces, not only to the lives of people around the world, but to economies around the world that have been subject to shutdowns as a result of programs to try to forestall the impact of COVID-19 on countries. In order to have impact, a vaccine has to be used. And that's what vaccination programs are for. And before the vaccination programs, we have to ensure that there's both a supply of COVID vaccine and a demand for COVID vaccine. And finally, we have to work with developing country manufacturers or manufacturers around the world to make sure that vaccines can be made. What are shown in the orange boxes are the other additional steps. We need the vaccine manufacturers. We need to do clinical development. We have to do the kinds of clinical trials that will show that the vaccine is safe and effective. We then will need to seek approval from a Ministry of Food and Drug Safety or a Food and Drug Administration. We'll have to achieve WHO pre-qualification if this vaccine is going to be used in parts of the world that don't have strong regulatory authorities. So in sub-Saharan Africa or parts of South Asia, World Health Organization's approval is important in order for UN agencies to purchase vaccines for Gavi to distribute. And so you can see that in the next box. We also need a recommendation from the WHO Strategic Advisory Group of Experts in Immunization. They will provide an actual recommendation to use the vaccine for countries that don't necessarily or can't necessarily constitute a qualified National Immunization Technical Advisory Group, or NITAG, the last item in the box under WHO SAGE. So all of these organizations make a recommendation for the use of the vaccine. That creates a demand. The vaccines are purchased and used. That's the vaccination program. And then ultimately we have an impact. How does IVI and how do our global partners collaborate in this? You see that in the blue boxes. We form product development partnerships that are international and cooperative and collaborative. We work with academic laboratories, with companies, with the WHO, with Gavi, in order to create the supply and the demand for the vaccine. So when we look at COVID-19 vaccine development, and this is a picture, if anyone's been to Rome, of the Spanish Steps. AC means after COVID. And you can see they're empty. And when you probably visited Rome, the Spanish Steps were probably, as they are in the second picture, BC, before COVID, packed with people. In order to go back to the future, we are going to need a COVID-19 vaccine. And the three elements then, to summarize, are to prove that the vaccine works and is safe, to make the vaccine with high quality and in sufficient quantity, and finally to use the vaccine to prevent infection and disease in a manner that provides equity and access to people around the world who need a SARS-CoV-2 vaccine. Thank you.